Thank you, Chairman, for inviting me, and thank you to all of you coming to listen to us this morning. As Stefan said in his quite thorough and interesting talk around some of the molecular and translational scientific developments, the context matters. So what I will try and do, for those of you who are not deeply embedded in oncology as it is practiced, is to set the context of what is happening in oncology so that we can then see where innovation may be needed. The bad news is that cancer is getting more common. In our lifetime, half the people in this room will develop cancer. Now, that's not meant to scare you, it's meant to be a reality that we face. And about half of that increase in incidence, just over, depending on which cancer, is due to the fact that we are better at treating other diseases, so we live long enough to get a cancer. But that's also a challenge because it means that the age of the patients we treat is also increasing. The workload in our cancer hospitals continues to rise. I can't comment about surgery, I'm not a surgeon. But I know in many hospitals, in the day chemotherapy units, the annual increase in patient attendances is between 5 and 10%. We don't have a 5 or 10% increase in staff every single year. And the reasons for that is that we are better at treating patients with advanced cancers. Fantastic. We have new drugs, new approaches, the patients live longer and get more treatment. We are also treating more patients with early cancers, with drugs as well as surgery and radiotherapy. And, of course, the whole volume continues to increase. The second reality is workforce. Clinical staff, whether they're pharmacists, nurses, radiographers, doctors, we are not cheap to train. It's several years of undergraduate, postgraduate training. You cannot turn on a workforce in 1 or 2 years. We need to plan 5, 10, 15 years ahead. And in many countries, workforce planning is not very good. And increasingly, and I'll give a couple of pointers to this later, patients want and are right to want to be treated by an expert workforce. They expect us to know more and more about the diseases we're treating, and that takes time. And of course, our economies are not behaving the way we want them to. So we have a difficult challenge. More work to do. Most of it is good stuff for patients, not necessarily enough people to do it, and not necessarily enough money to do it either. So let me paint 2 very quick scenarios, and they're meant to be somewhat of a caricature. 20, 30 years ago, as an oncologist or a surgeon or whatever, I might treat many different kinds of cancers. I would make my decisions on my own. I might occasionally ring a friend. And I would follow up my own patients. I would have this little empire of my patients, and I'm the best person to know what to do with them. And it probably wasn't very difficult, because with 1 or 2 exceptions, there were very limited number of treatments. It's not difficult to know how to use 6 drugs. It's more challenging to know how to use 60. And now, rightly, we've moved to multidisciplinary teams where you may have 5 or 10 different experts making a decision in one disease. Instead of one person making a decision in 10 diseases, you have 10 people making a decision in one disease. So, for example, in my own institution, we see 700 new breast cancer patients a year. We've worked out that we have the equivalent of 1 whole-time senior clinician in MDTs every week of the year. That's a resource that is well placed, but it's expensive. And of course, we have many new treatments with many newer toxicities, cardiovascular, immunological, and of course the risk for interactions. So the system is getting more complicated because of innovation. Stefan, this is a coincidence. I hadn't seen Stefan's slides. Stefan looked at something, the revolution at a molecular level in lung cancer. When I started in oncology, the the question that was asked about lung cancer was, is it non-small cell or small cell? And that was fundamentally your treatment distinction. There was modest benefit from chemotherapy. A small proportion of small cell patients with limited disease could be cured. Most radiotherapy was palliative, and a small number of patients could have curative surgery. It wasn't easy to treat lung cancer, but the spectrum of what you had on offer was fairly limited. Now it's completely different. You need a multidisciplinary team. You need to understand the EGFR mutation status of your tumor. You have a series of drugs in that space. We now need to know about the ALK status, and we have crizotinib as first line. There are second line drugs coming in. We have a range of chemotherapies, just getting approval for the anti-PD-1 drugs. Even lung cancer has got a lot more complicated, and yet we still have yet to make dramatic differences to outcomes there. But there are innovations away from the molecules that need to be made. So, coordinated approaches to care. If I see a patient, do I get every single piece of information about that patient, even in my own country's healthcare system? No. I get much of it. I don't get all of it. There's still a lack of accurate and precise information sharing. Multidisciplinary working. Is it absolutely standard for every cancer patient across Europe? No. Why not? Before we're worrying about next generation sequencing of every tumor, what about multidisciplinary assessment of every patient? Quality approaches to diagnosis. If I get a pathology report from another hospital, do I know that that's correct? Well, depends which hospital. Depends what quality assurance process is in place. Is that standard across Europe for every molecular marker? No. And there's a strapline which has come through in the UK, and I can't translate it to all the languages, but patient involvement is key. We used to be very paternalistic. I think you should do this. The strapline that's come through is, from a patient perspective, no decision about me without me. You don't make the decision, we make the decision. Is that universal in Europe? I don't know, but probably not. So we already have innovative approaches to cancer care that we have not completely implemented. So that's at the system or systematic level. Obviously, at the individual patient level, we have a number of important developments. Breast cancer is the disease I know most about, so I tend to talk about it because my knowledge of other areas is shrinking. So we measure hormone receptors in breast cancer. I can tell you in the UK, that was a battle to become standardized because one or two key opinion leaders said it was irrelevant. Now, I think pretty well standard across most of the world where it's possible. HER2 testing is pretty well standard in breast cancer. It's coming through in gastric cancer. And there is a message of context. Because HER2-positive gastric cancer doesn't have the same implications as HER2-positive metastatic breast cancer. You may use the same class of drugs. They may be effective. But the natural history of the diseases is not the same. So the context, even for one molecular test, matters. RAS status for the use of EGFR inhibition in colorectal cancers is becoming standard. It's the wild type that matters, not the mutation. And, of course, for inherited genes, BRCA1 and BRCA2 testing, important for a patient to know their risk of developing cancer. It's also becoming important for knowing how to treat the cancer. How universal is that across Europe? And importantly, are the guidelines for getting the tests the same? Nope, not even in my own country. So I can see a patient in Edinburgh who we will get a BRCA1 test on. In another city in Edinburgh, the geneticists say, nope, her risk isn't high enough. The risk is irrelevant. She has breast cancer. This is a test about how to treat her. So even in one country, we don't have a universal approach to making available stuff that is now relatively straightforward. And of course, we're moving into an era of multi-parameter tests. Again, I quote the breast cancer ones. These are prognostic, primarily. They may have some predictive relevance for use of chemo. At least 3 out there, Oncotype, Mammoprint, PAM50. They will come through in other diseases. Are they universally used in the same way? No. Now, Stefan gave a very good molecular basis of personalized or precision medicine, but you need to know what you're looking at. So, sorry about this. These individuals all have the same gender. They are all wearing the same headgear, both in terms of shape, color, and probably manufacturer. They're all carrying the same thing in their right arm, an umbrella, essential in the UK. And they're all wearing a dark business suit. You need to know where to look. The difference is, of course, whether they've got stripes. Take a different set of people, and you need to look at a different characteristic in order to personalize them. You can't just say, this is the approach that works. So for some tumors, mutation sequencing matters. For some, it's copy number variation. For some, it's protein expression. And I'm sure Stefan is right that for others, the microenvironment, the tumor niche will matter. We don't yet know which one needs which approach. And if we look at the wrong thing, they all look the same. And we miss that opportunity for a personalized approach. But personalized or precision medicine needs to be achievable. If it involves 16 different biopsies of a patient with metastatic cancer, is that really deliverable in every hospital in Europe, let alone the world? It's not going to be. There are patients for whom biopsies are more dangerous, either because of their comorbidity or because of the location of the tumor. It's much easier to take a biopsy from the surface than deep in a viscera. And we need to know how to use the data. So a precision approach has to be achievable, not necessarily in every patient, but across the board, to be implementable. It has to be affordable. And we cannot escape the fact that the costs of delivering healthcare are rising faster than the resources made available to deliver healthcare. So whole genome sequencing used to cost millions of dollars for one sequence. We can now do the wet lab sequencing for around about €1,000 plus. So in theory, it's probably the cost of doing a PET scan. So in theory, we could sequence every single cancer in Europe. What we don't have are the trained bioinformaticians and clinical geneticists to interpret the data. And they, I'm afraid, will cost an awful lot more than just a few more €1,000. So we can do some of the technological developments, but have we got the system in place and the trained people to interpret the data? And Stefan's hinted at this, and I think this is also important. The cost of drugs is rising faster than inflation. Are they worth that extra cost is an important question. And I think personalized care has to be acceptable. It's not just for the young, who are fit and can deal with the additional tests, and it should not just be for the rich. And when we deliver a personalized approach, the efficacy, there's a strange word in there for those who are not English speakers, don't use the word outweigh. It's not one I recognize. It's my typing. It wasn't a deliberate joke. The efficacy has to justify the toxicity. Just 2 more weeks in progression-free survival for toxicity may get your drug to the market, but actually are you giving what patients need? Now, this is an example of why the regulators, to my mind, need to be part of the conversation. So again, HER2-positive breast cancer, metastatic, and previously treated with Herceptin. GSK had a drug, now called lapatinib, which looked like it worked in this situation. So they go to the regulators, the FDA, because they're based in the US, and say, we want to demonstrate efficacy. And they said, well, the only approved drug in this setting is capecitabine monotherapy. So combine your drug with capecitabine, show it's better, and we'll give you a license. And that's what they did. And I was one of the 2 CIs on the trial, so I'm conflicted here. Left-hand side, add lapatinib to capecitabine, and you get better control of the disease. On the right-hand side is a trial run in Germany, same control arm, capecitabine, but what they did was what we were doing in the clinic, which was adding Herceptin to capecitabine, because we had some phase 2 data to suggest it works. The axes are different, and I tried to fiddle around getting them to look the same, but it distorted the graphs. Add Herceptin to capecitabine, and it also works. So which one do you think we use in the clinic much more? The right-hand one, because it's less toxic. But the regulators were not interested in doing a comparison against the real world, because it wasn't in the regulators' world, because it's not an approved therapy. And if you look across cancer, you will find other examples of where a regulator's approach to the right trial to prove efficacy is not actually reflecting the question I want to know in the clinic and my patient wants to know. They would like to know which of the two superior treatments is better. I know the answer in terms of toxicity. I don't know the answer in terms of efficacy. This also caused a headache for health technology appraisers because, of course, the health technology appraisers could only compare against capecitabine monotherapy because that's what's in the trials. So the regulators here were imposing an approach which worked as a scientific experiment. Yes, it's better, but actually didn't address the real question in the clinic. So reimbursement is a challenge. When, I notice I put the word when would it be appropriate to spend 10,000 euros a month on a drug? The answer is maybe sometimes it is. But when I started in oncology, people were complaining about drugs that were costing 100 euros a month. So you can see the price inflation has gone way up for lots of reasons. I'm not challenging the reasons, I'm asking the question is, when is it right to spend that much on a drug? What benefits do that drug actually bring? The clinical trials are a carefully designed, usually very well conducted experiment. Compare A with B in that setting, in those patients, with those precise criteria, and you get an estimate of the benefit and an estimate of the toxicity. Take that to the real world, and it may behave very differently. Sometimes it turns out to be a lot more toxic. Sometimes it turns out to be a lot more efficacious. Herceptin, arguably, in metastatic disease, is probably better than the trials estimated. Fortunately, we have access to it. And if a patient comes to me and I say, you should have this treatment, how sure am I that he or she will get those benefits? Because, in fact, that patient would have some particular aspect which would have meant they were ineligible for the trial, even had it been running. So do I know that that result translates to this broader population when I don't know their comorbidities, their concomitant medication, and I certainly don't know their genetics? So what other innovations can we do? And Stefan's hinted at this, and I think there is a big opportunity across Europe. I suspect most hospitals prescribe their chemotherapy electronically. All will prescribe their radiotherapy electronically. In many parts of the world, you're prescribing in the community from your general practitioner. Even if the prescription isn't electronic, the pharmacist will have an electronic record. We'll have electronic hospital records, and we can, but rather intermittently, we can collect patient-reported outcomes electronically. They can do it on a smartphone, they can do it over the web, they can do it sitting in clinic. We could capture an enormous amount of data about what we're doing today electronically. And yet, most of the time, we don't use that. We may do specific audits, but we don't look across the whole picture. And why do I think it matters? Well, actually, how do we treat patients now? Are we using our current therapies in the optimal way? Probably not. How well do those treatments actually work? Do they deliver the benefits they're supposed to? Patients ask us, and unless we've done an audit, we don't know the answer. And if we're coming in with an innovation, in the trial it was compared with drug Y, which is the approved medicine in the US. Actually, the Europeans may completely ignore that or may not always have access to it. So in fact, what would that drug replace? What is happening now in that population of patients? And would we use it in the same sequence? Does the sequence of drugs matter? Probably, but not always. So, last couple of minutes. Where next? The first innovation, I think, is to do what we can already do better. There's an awful lot about what we know works in cancer care that is not standard across Europe. Multidisciplinary working, specialization, etc. There's also stuff we all do, which in fact is probably a waste of money, time and resources. So, breast cancer again. There's very little evidence that seeing your doctor once you've been treated for breast cancer has any difference on your outcome, and probably very little difference on your quality of life. And yet, in different ways, in different places, we do different amounts of follow-up. Why? Because some people think it's a good idea. Actually, it's probably a waste of resource. Research is good. That's fine. There's data that if a hospital is research active, all the patients in that area benefit. Not just the ones in the trial, 3-5%, but the whole cadre with that disease. It's true for ovarian cancer, colorectal cancer, cardiovascular disease. So why do we not embed research in our hospital healthcare systems, and indeed our primary care health systems in Europe? Because that would improve outcomes. Research is seen by some as a special, extra sort of luxury. It shouldn't be a luxury. It should be there for everybody. So measure what we're doing now, and we might be surprised. Now, this came out coincidentally in The Lancet very recently. I haven't put the whole editorial up, but I think the top heading is really important. This is looking at a global perspective. Access to medicines. The Lancet is arguing the status quo is no longer an option. There needs to be a change in a system so that people worldwide can get access to medicines. I think there are other innovations that we could already do that we're not putting in place. We need innovation to implement previous innovations, whether they're medicines, multidisciplinary working, whatever it happens to be. And finally, who are these stakeholders who need to be involved in this conversation? In the drug approval world, it probably feels to many clinicians that these are the two parties that matter. And yes, they're important. The regulators need to approve, and those who develop drugs need to develop them. But actually, health technology appraisals is becoming critical. If you have a fantastic drug that costs £1 million a day, not many people will get it. And actually, you need to build into your trial designs, I would argue, or your post-trial designs, a measure of just how good that innovation is. Very often, the model by which a drug comes in through the regulatory process doesn't fit with what people do in the clinics. So I think we need a bigger circle here to involve those who do the treating. And yes, there's a gap in the middle, because sometimes it feels that's what's happening. That's for whom all this is about. We need to put them back in the center and think, what is it they need to know? What's the information that they should be made aware of that can be gained from many sources to implement innovation? Thank you.